Hello, everyone. Welcome to the latest Novogene webinar, RNA Sequencing Results Explained, what you can expect from the analysis. This is our second webinar of 2022 and is the companion piece to a beginner's guide to RNA sequencing that we held in January. You can, as ever, find all our previous webinars on our website to watch on demand. I'm delighted to say that our beginner's guide webinars continue to grow in popularity. We have over 1,800 people registered for today's webinar. If our webinar can help just one of you achieve better sequencing results in your science, we'll feel it's all been worthwhile. On that note, we would love to hear your feedback, particularly around how we can make these webinars more useful to you. We'd appreciate it if you could take a couple of minutes to complete the survey that will pop up after you leave the webinar. Bit of housekeeping before we start. The webinar will run for a total of 1 hour. There'll be around 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote questions by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. But don't worry, if we don't have time to answer your question live, we will come back to you via email after the webinar. So please don't use the raise hand functionality. Just send us any questions via the Q&A or message us directly for the technical issue. We will share the slide deck and a recording of the webinar via email. You can expect this on Friday at the latest. Due to popular demand, we have added a webinar attendance certificate for any students on the webinar who require one. There'll be a link in the follow-up email to get a certificate that you can save or print. Well, I'd now like to introduce your host for the webinar, Rana Jerdek, who is one of our French account managers. Rana, over to you.
Thank you, John, for the introduction. Good morning, everyone. I hope you're all doing fine. Thank you for joining us today for our webinar entitled RNA-seq Results Explained, what you can expect from the analysis. My name is Rana Jerdek, and I am the account manager for France. And as my colleague John said, I will be your host for today. So without any further ado, I will start by giving you just a short presentation about our company before handing over to Dr. Shiki An, who is our product manager and who will go into more detail regarding our webinar's main objective. So our Novogene was founded in 2011, and we have been growing very rapidly ever since. Our mission is to advance genomics and improve life research with a specific field dedicated to the clinical research in order to improve diagnostics and human healthcare. As you can see here in this picture, our work covers many aspects of the next generation sequencing with a wide variety of applications. Our European Next Generation Sequencing Center is located in the Cambridge Science Park campus in the United Kingdom. Here is a picture of our state-of-the-art ISO-certified laboratory, which is equipped with the most recent Illumina Nova 6000 sequencers. We even have our own supercomputing cluster, which enables us to offer you bioinformatic analysis and publication-ready data. We are even able to store your data in the UK for further use. And here, I'd like to invite you to take a virtual look of our UK lab by scanning this QR code or by visiting our website, www.novogene.com. So since its inception, Novogene has established many operations and laboratories across the world, serving more than 60 countries across 6 different continents. We offer services across Asia, the United States, and of course, Europe. And in order for us to do so, Novogene has carefully selected a professional team of over 2,000 employees to meet the requirements of more than 6,000 customers from world-class universities, biotech companies, and hospitals. We are able to handle small and very large-scale projects. So what we can offer you is our our wide range of sequencing services that cover a full range of applications, varying from the human genome sequencing to the transcriptome, the microbial genome sequencing, the epigenome, and the animal and plant genome sequencing. So for today's talk, Dr. Chichi will concentrate on the bioinformatic analysis of the transcriptome. So we all know that the next generation sequencing market is very crowded today. So why should you choose Novogene as your sequencing partner? Here is a short summary of our core benefits, but I would like to take advantage of this slide to actually share my own experience with my clients. As a matter of fact, what my clients are mostly happy about is that when we share when we send them an email with the confirmation with the statement confirmation, so they can see partial results and confirm that everything is okay with them, I instantly get emails back saying that the data quality is excellent. When can we have access to the full results? So this is something they really appreciate. Another example I'd like to give is that once we had an RNA sequencing project for over 100 samples, and the client needed a short turnaround time of 3 weeks because she had a student that was staying with her for a very short period of time. So we were able to do that for her, and she was very happy with the outcome. Another thing we are very proud of is our record in the scientific committee. As a matter of fact, we have been cited in over 590 research papers that were published in elite journals such as Nature Genetics, Science, Nature Communications, and many others, with a total impact factor of over 6,120. We are even driving the next generation sequencing industry forward with our own next generation related patents. Another proof of customer trust is our ever-growing Novogene community. As a matter of fact, our CS team, which is located all across Europe, is growing on a daily basis, and we are all very happy to help, or very delighted to help you with your project. So here is a picture, or I'd like to introduce to you our sales team. We really live next to you, we speak your language, and we just want to help you get the best out of your projects. Here you can see the UK and Ireland team, the Dutch team, the Nordics team, without forgetting the Southern and Eastern Europe teams. And here I'd like to take advantage to encourage you to contact your sales representative if you already know them. If not, you can fill an inquiry on our website, www.novogene.com, or you send us an email at info@novogene-europe.com, and the appropriate sales manager will directly get back to you with an answer. And with that, I'd like to thank you all for your attention, and I'll hand over to my colleague, Dr. Chiki, who will go into detail about today's explanation, about today's webinar. So thank you for your attention.
Hi, Rana. Thank you very much for the introduction. And then just bear with me for a minute to set up the slides. Okay. Okay, just bear with me for a second. Okay, so before I go into further details about the webinar today, I just wanted to highlight, as the title suggested, this is a beginner's guide, and we're not going into any details about how to coding, exactly, the details of the bioinformatics. But what you're going to get from this webinar is to understand through those data you sequenced, what information we'll be able to get, or how to interpret those sequence results. Okay, and so to start, I'm going to give you a very brief overview about the content we're going to cover today. So first, I'm going to briefly talk about what is RNA-seq and the solutions and Novogene offer for RNA sequencing. And then I'm going to use a eukaryotic mRNA sequencing project as an example, and to go through the bioinformatic analysis and workflow. And this is actually from a quality control to a characterization of transcripts analysis. And in the end, I'm going to briefly talk about other BI services we offer for RNA-related RNA sequencing services. And then in the end, I'm going to have a very quick summary to recap what we talked through today. Okay. And so what is RNA? For the audience here, probably all have heard this. So RNA-seq is basically RNA sequencing. This is a technique utilize NGS and next generation sequencing to reveal the presence and the quantity of RNA in a biological sample at a given moment, which will allow the analysis of continuously changing of cellular transcriptome. So it depends on what, as a research purpose, what is the aim of the projects, the biological question we want to answer. There are two common applications for RNA sequencing. So the first one is qualitative analysis, characterization of transcripts. So this can be used for a genome annotation or identify orientations of transcripts, importantly identify alternative splicing or study gene fusion, transcript fusion. And then sometimes also can be used to study RNA kinetics as well. And the other major application for RNA sequencing is quantitative analysis, basically look at expression. And so this can be used to identify differentially expressed genes and the different conditions tested, and also can be used to identify isoform expression level as well. Okay. So our bigger picture, so what those information can be used for, what type of biological questions can be addressed from RNA sequencing data. So one major application is to identify biomarkers. And then another area is to look to study host pathogen interaction. And also for microbial community study, RNA-seq can be used for mitotranscriptomic analysis as well. So if you're interested in single cell resolution or identify a cell type and a subtype characterization, so RNA-seq can also be utilized at a single cell level. And of course, there's a lot more applications for an RNA sequencing. Okay. So it depends on what is, again, your research aim, your project. Okay, there is different solutions Norwegian can offer for you. So if you're interested in to just study gene expression, okay, and depends on what type of an RNA you're interested in. So there's different options here. So if you're interested in mRNA, then there's mRNA sequencing and we offer, and then we also offer a long coding RNA sequencing as well. For circular RNA, we have circular RNA sequencing service and also a small RNA sequencing service as well. If you're interested in all of them, and we also offer a total RNA sequencing service for our clients. Okay, and if you're struggling with your starting material, okay, and then we actually offer a ultra low input RNA sequencing service. And if you're interested in single cell resolution, then pre-made library would be your choice here. So if actually you're interested in de novo transcriptome, it depends on your the quality you want. If you want a reference quality transcriptome, then it's actually RNA-seq based on PacBio would be your best choice. But if you don't really need a reference quality transcriptome, then long coding RNA or mRNA sequencing will be sufficient for you. Okay. So if your project is really just looking into alternative splicing or isoform analysis, then the full-length transcriptal sequencing, it would be your best choice. And this will be PacBio RNA-seq based or an Oxford Nanopore based sequencing project. Okay. So besides those, we also offer a couple other RNA-seq services such as exosome RNA sequencing and mitotranscriptome profiling and duRNA sequencing. Okay, so if you're interested to know more, so please visit our website for our previous RNA-seq Beginner's Guide webinar. So for today's webinar, the major content is actually mentioned as to utilize mRNA-seq project as an example to go through the analysis workflow. So this will include 5 major steps. So to start from a quality control, at this stage, basically your raw data will be filtered and generate a clean data set. And this will be utilized for used for the downstream analysis. Now, once you have your clean data, and then the next step is basically mapping your clean data to the reference genome, the mapping. And once these steps are done, it's basically looking for expression analysis, identify differentially expressed genes. So once you have this gene list, then you wanted to assign biological function to those genes you identified. Okay, that is the step 4. And on the other side, actually, MR sequencing data can also be used to study and to characterize transcripts. Okay, so this including variation calling, alternative splicing analysis, and also fusion transcripts analysis. Okay, so in the next few slides, I'm just going to go into a little bit more detail about each individual step. So we start from quality control. As I mentioned, we start from raw data and end up with clean data. So how the raw data look like? Okay, so raw data, it's usually will be stored in FASTQ files. Okay, so this is generally how it looks like in a FASTQ file. So it contains 4 lines, and then each line contains different information. The first line contains information about the sequencing ID and also the library index. It's here. And then the second line is the most important one because this is actually the read that is sequenced. And then there is a separator symbol here. And then you will also have a quality score. So this score basically indicates what is the sequence quality of each base that has been sequenced. Okay. If it is a pair and run project, there will be a second file with the 1-to-1 corresponding headers and reads. And actually, each file, FASTQ file, has millions of reads. So this is just one read. So the file itself can be really large in size. And that's why almost all the time, the FASTQ file will be compressed. And then the file will become a FASTQ GZ file. So that's just a compressed FASTQ file. So another part I wanted to just briefly mention is the quality score. So you probably have heard this or seen this previously. It's the Q30. What does this actually mean? That means for error probability, that means potentially there's 1 in 1,000 base you read potentially has error. But also that means in the other way, for accuracy, that means 99.9% is actually accurate. Okay, so that is the quality score means. Okay. And so for this step, quality control step, we use a software called FASTP. As I mentioned, the input is raw reads, it's a FASTQ file, and the output is clean reads, and it's still a FASTQ. So what you're going to obtain from this step, basically you will have a quality report, which including the error rate of each base. So basically you can see the error rate of each base at each position to make sure the quality of the sequence is good. And also you will have the base content information as well. Again, that's the content at each base that is sequenced. And importantly, there will be a data filtering steps here, and then that is the step really clean up the data set. So here is going to remove the reads containing adapters, and then the reads containing ambiguous readings, and also reads with low quality as well. So you'll end up with a clean data. So this is just shows the quality of our sequence is really high. So it's only a very little proportion reads will be filtered away. Okay. So now we have a clean data in hand. We're going to move on to the mapping stage. At this stage is basically map the reads to a reference genome. So this step is really just determining the location from which each read is actually oriented. So it is possible to map the reads to reference genome, known as genome mapping. And this is by default and the most broadly used approach. But also you can, if you like, to map it to transcriptome as well. And so for RNA sequencing results mapping is slightly different to standard DNA mapping. So why is this? The reason for this, you can see here, there's 3 different types of reads. Okay, the ones in red, that's very straightforward. Okay, it maps entirely to one exome. You can see here. And for the blue and the purple, actually they span over 2 or even more than 2 exomes. Okay, here. So that will cause a problem when it comes to mapping. So for RNA sequencing mapping, it requires a mapper called a spliced mapper. Okay. So this is the principle how it actually works. So the first, it will be try to identify the reads that only spanning single exome. Okay, so this first step, and then the ones that mapping to other splicing junctions of those unmapped reads will try to map again. So this is the major difference when it comes to RNA-seq mapping and DNA sequencing mapping. So the software we use for this step is high-seq2. So this is the full name of this software. And of course, there are other packages that is available, but there are paper or publications have shown that this software generate more accurate results and with fast and sensitive alignment. So at this step, what you required is the clean reads we generated from our previous step and a reference genome, and that will be in FASTA format. Okay, so the output we're going to get is the BAM file. BAM file is a binary version of a SAM file. SAM file stands for sequencing alignment map. Okay. And then besides getting the mapped results, a BAM file, you'll also receive a summary of the mapping results, as you can see here. So what is included in this table here? It has the sample names, and also it has information about the total reads, how many of those reads are mapped, and how many of those reads are actually uniquely mapped. And then also the percentage of those mapping results as well. And then this results, basically, you have them across different samples. So this is very quickly, you can see, how good the mapping results it is. Okay, so once you have your BAM file, actually you can actually use software to visualize and inspect the results already. So one of the most popular used platform is IGV, Integrative Genomics Reviewer. Okay, so at this stage with the BAM file after mapping, you can already see the position of single or multiple reads in the reference genome. As you can see here, they are basically mapped reads to a certain location of the genome. And then you are also able to see the read abundance of different regions to demonstrate their expression levels. Okay, so you can see the peaks are higher, and those ones, it means they have more reads mapped to that region. Okay, so very straightforward. And so later on, after next step, actually, you will be able to add an annotation tracking as well. So after that, you will also be able to see annotation information along with your sequence results. And also you will be able to see how those reads distributed between exons, introns, and intergenetic regions as well. Okay, so this would be the annotation track. Okay. So now once we have mapped the sequence results, we're going to move on to the next stage and trying to identify differentially expressed genes in our study. So how we're going to do this? So very straightforward, you would think, if we can count those reads, if they have more reads, generally, it means it's CRES higher. Okay, and actually that's exactly what we do for RNA-seq analysis. Okay, basically in this step, we're going to count the number of reads that is actually mapped to exon or genes. And so the software we use is feature counts. Okay, so in this step, it will count the reads that map to a single location, which means uniquely mapping. Okay, and then we'll assign those reads to a gene or exon. So here, what you require to have is a BAM file. So that is the file we obtained from our previous step. And then you're going to also need an annotation file. Usually it's a GTF file. And then, for this step, output is very simple. It's basically a table. It is a count matrix. What it has is you have the gene name here, okay, and then you're going to have the reads count for each gene, okay, here, and then this just means different samples. Okay, so basically you have reads for each genes from different samples in this count matrix. So now this data can be further used then for expression level analysis, but actually it's not that straightforward. In the next report, I'm going to explain why we can't really just use directly the row counts for further analysis. So before I go anything further, I just wanted to get one concept to come across. So it's the concept about coverage. So in next generation sequencing, coverage basically describes the average numbers of reads that align or cover of a known reference basis. So the 2 dimension of this. Okay, so coverage could mean depth of coverage and can also mean breadth of coverage. But for most of the cases, when we talk about coverage, it's depth. So basically, it means how strong is a reference covered by the sequence fragments. So sometimes it's expressed as, I need 30 times coverage or 2 times coverage. So this is basically means the depth. Okay, so just bear in mind this concept because we're going to use it in the next part. So here, why we can't just use directly the row counts for analysis, because the expression, okay, not only depends on how many counts, it's actually depends on the total read sequence we obtained from the sequencing. Basically, the depth matters. So here's an example to show why we need to take this into consideration. Okay, for example, there's a treated sample, there's a control sample here. For the treated sample, the sequence depth is 5. You can see it's covered 5 times. And then for the control sample, you can see it covered twice, basically the depth is 2. And for the first one, we have 2 million reads, total reads. And for the control sample, we have 10 million reads. If we want to compare, which sample has the higher expression, or has more transcripts. Okay, if we say 50, so this one's more, but actually that's not accurate. If you take the depth into consideration, okay, so depth normalized to 2 million reads. Okay, so this is only equals to 0.1. However, for this sample, if you normalize the total sequence you obtained, this is equals 0.2, which actually means the treated sample only has half of the transcripts expression of the control. So that's why I explained why we can't just use the raw counts for estimation of expression. Okay. Now, this is really between 2 different samples, but how about within one sample but different genes? So this means, okay, the read counts also depends on the total gene or the transcripts length as well. Okay, this is basically within sample, but the gene has different lengths. Then this is another example. So you have gene ABC, and then they have different lengths 2, 1, and 3 kb. And then for gene A, you have 4 reads mapped to there. And then for gene B, you have 5, gene C, you have 9. Okay. But you can see if your gene is longer, then automatically it have more reads mapped to it. But that doesn't necessarily mean it express higher. So it will need to be normalized to the gene length. Okay, so after normalization, actually you can see that actually gene B has a highest expression level. So this just shows it's important to normalize the raw counts before we use them to estimate expression. Okay, so there's a couple of different ways to normalize RNA-seq data. So the most well-known one would be an RPKM and FPKM. So RPKM stands for reads per kilobase of exon per million of mapped reads. So this is really useful for single-end normalization. So for FPKM, so the difference is here is fragments, because this is the one used for paired end read normalization, but they are really, really similar. It's just for FPKM. So this is tracks of actually of fragments. So one fragment with 2 reads, okay, then it only will be counted as 1. So that's the major difference between the two. And you also have a TPM, so transcripts per kilobase million. This again is very similar to the other 2 method, except the order is different. This will be normalized the gene length first, and then it will normalize the gene sequence depth as a second. Okay? And this method can be used for either single or paired end reads. After this step, then the counts can be used for as a measure of relatively abundance of transcripts. Okay, so at this stage, actually, after you have obtained those information, you'll be already very quickly to see the intra and intergroup sample variability and the outliers already. So there are 2 different ways to do this. So you can have the Pearson correlation analysis between samples. As example here, you can see if the samples are really similar, expression are really similar to each other, then the R-square is going to be high and the color will be darker. Okay, if they were different from each other, Okay, the R square number will be low, and then the color will be lighter. So you can see here, you can see the samples from the same group. The replicates between each other are really dark. It means they're really similar to each other. The reproducibility of your experiment is good, but between the samples, the treatment and control, you can see there's a clear difference there. Okay, and another way to look at this is through a principal component analysis. Again, this is a very quick way to see, how good the reproducibility of your experiment or samples from replicates or from same group. You can see they're really nicely clustered together. That means the reproducibility is good. Okay. And then now we're going to talk about how we actually identify the differential express genes. So the software we use here is DE-seq2. Okay, and I won't go into too much details about how exactly the software runs, but I'm going to just give you a explained principle about DE-seq2. So this is basically provide a method to test for differential expression. So what DE-seq does is actually it's going to calculate a group mean. There, the pink circle here. So for sample A, group A, and then for group B. Okay, so you have two group means, but it will also calculate the global mean. So this is the yellow dot, and then it's going to calculate durations of the group mean from the global mean. So if they're really separated, this will be considered a significant difference. If they're really close together, that means there's no significant difference. So that's the rationale, the principle behind DE-seq. Okay, so for this stage, actually is require a row counts as the input, and then output will be a result table. So why is only required for the row counts? Because DE-seq is going to have this own built-in normalization already. So the first step DE-seq does, it is normalization. Okay, and then the next step is going to estimate for fold changes. And then for the last two steps is basically statistical analysis to test if those changes are statistically significant. Okay, so basically that's hypothesis testing and then to adjust to the p-values. Okay, and of course there are other options, alternative options such as AGR we can offer for our clients. If you're interested in to use the other approach, that's fine as well. Okay. And then, so we talked through the principle, and this is the basically results table, how it looks like. Okay, so what you're going to have here is that you're going to have a gene ID, and then you're going to have the fold change calculated for you. So basically, this is group 1 or sample 1 versus sample 2 versus group. So basically, the comparison between them. Okay, and then you're going to have a p-value and a p-ADJ value calculated as well, and to check if they are significantly a difference or not. And also you're going to have annotation information about gene, each gene that's identified. Okay, their description and their location, where they are on the genome, basically. Okay, so now after this step, you're going to have a gene list that contains the genes that differentially expressed and different conditions you tested. So how we're going to visualize this data? So there's a couple different ways here. So one of the very popular method to visualize this is through a volcano plot. You probably have seen this before in many publications. So this is really good when you look at two different groups or two different samples. Okay, so the green ones here are the genes that's downregulated, and they're significantly downregulated. Okay, so this is the cutoff and the threshold. And then the red ones here are the ones that upregulated, and then again there significantly changes between conditions tested. Okay, and then those blue ones will be considered there's no significant difference between the two samples or the two groups. Okay, so but if you have actually more than two samples or two groups, so there's other ways you can visualize this. This is through cluster analysis and differential expression. So here you can see the three samples on the left, they're from the same group, and then the three from on the right hand side from another group. Okay, and you can see the samples from the same group cluster nicely together, which means the reproducibility are quite good. And also different color means a different expression level. The blue here means high expression, and the red ones means low expression. And you can see between the two different group, the expression pattern is very different. Okay, so it's very quickly, to see the reproducibility and then the different expression pattern between groups and samples. Okay, and so there's other ways you can look at your differential expressions. So this is through a one diagram. Basically, here you wanted to look at the co-regulated genes. It depends on the experiment setup. Okay, you can have different type of one diagram here. Okay, although they look very similar, but actually the meaning it expressed, the information is actually contained is quite different. Okay, so here for the first experiment setup, for example, you have group 1 and group 2, and you have a control. Okay, and then at here you're going to compare group 1 to control, and then compare group 2 to control, and you end up two sets of differentially expressed genes or DGEs. Okay, and then you wanted to see if there's any overlap between those two sets of DGEs you identified. So, and this is the one diagram. So usually what is the situation this can be used is, for example, you have wild type and then you have two different type of mutants, and then you wanted to see at a regulatory scope and there is any overlap or difference between those two mutants. So this is one of the situations you can use this type of one diagram. However, there's more complicated situation. So for example, here, so you have a group 1 that has an existing drug. Okay, it's already have and it's used in clinical, and then through experiment identified a few novel compounds. Okay, and also you're going to have a control group, basically that is uncontrolled and untreated samples. So what you want to do here is that you wanted to see, compared to your existing drug, how your novel compound performed. Okay, but before you do that, all the data needs to be normalized to the control first. Okay, basically you take away the background. Okay, and then you end up with 4 different set of DGs, and then you're going to compare that to the existing drug. Okay, to see how well or how different your the new compound performed at expression level. Okay, so basically it takes 2 steps. You're going to normalize this first to one control, and then you're going to compare again to one of the existing drug or one timeline or something. So this takes 2 different steps. So this can be, applied to different experiment designs. For example, for time course RNA-seq, or sometimes to an assessment of drug or chemical concentration response situation. So usually it requires 2 steps of normalization and comparison. Okay. So now at this stage, you now have a list of gene that has differentially expressed. Now we'll actually want to assign functions to them. Okay, and now we move on to the 4th step. Okay, so at this stage, as mentioned, is basically assign biological relevance to the DGEs. So here we're going to determine whether there is any enrichment of no biological function or interaction or pathways. Okay. And then we're going to identify genes involvement in those novel pathways or networks by grouping genes together based on similarity trends. Okay. And then the software we use for this is known as an unclustered profiler. So this is a really universal enrichment tool for interpreting an omics data. And the database, the most commonly used database, you can list here the Gene Oncology or GO database, and also CAG. The reason for this is because they are a long-standing curation, and also they're available for a range of species. Okay, so that's why there would be most cases would be the first choice. However, if you're studying human-related transcriptal analysis, then there's other anthropology and the pathway database that are available. So they're more specific. For example, there's this GenNet and the reactant pathway. So they also offer those database for analysis as well. Okay. So how this is going to look like, once after the analysis. There's a couple ways to present or visualize those data. So the first example is a geo-enrichment scatter plot. So what this figure means here, okay? So on the y-axis, you can see those are the geo terms identified. And then the dots, okay, so means the size of those dots means the number of differentially expressed genes identified, within this, under this geo term, okay. So the bigger, there's more genes and differentially expressed under this geo term. Okay, so the color means basically the PADJ value. So if it's actually significantly different or not. So on this one here, of course, they're all quite red, means they're all quite significantly differentially expressed. Okay, and for KAG pathway, okay, you can see, but basically differentially expressed genes where it sits in a pathway. Okay, so here the red means upregulated genes, and then the green means downregulated genes, and you can see clearly where it actually sits in certain pathways. Okay, so now after the function analysis, we're going to talk about a slightly different part, of the analysis as a quantitative analysis, basically trying to characterize the transcripts using an RNA-seq data. So the first one I want to mention is variant discovery. Okay, so though it's not very popular, but actually you can call variants from RNA-seq data as well. There is some advantage here. Okay, so this is really good. So for RNA-seq data, it's very good for calling SNPs and short indels. And then another advantage here is that you'll be able to identify the expression level and the mutation of the gene simultaneously from the same experiments. Okay, and then also RNA-seq in a way provided a better coverage than a whole exome sequencing, and then it is cost effective because they actually get into two informations from one set of experiments. Okay, the software we use is called Genome Analysis Toolkit, and GATK. Okay, so basically the input file will be a BAM, and the output file is called a VCF, is a variant code format. So basically, we're aligned reads identify where that aligned reads differ from the reference genome, and then write into the VCF file. Okay, and we'll also use a software package called SNPF, basically to give an annotation and a functional prediction of those mutations or variants we identified. Okay, and this is actually GATK compatible. So through the analysis, we're going to identify the variants and also have annotation information as well. Okay. Now, after that, we also can call alternative splicing as well. So splicing is a very important mechanism that regulate isoform expression in cell-specific or timing and specific manner. Okay, so alternative splicing actually generates tremendous complexity at transcription and proteome data, proteome level, most likely in higher eukaryotes organisms. Okay, and then any changes in those alternative splicing usually underlie gene regulation in diverse biological functions and also disease process. Okay. So here are some mechanisms or situations for alternative splicing, the different cases. So the software we use to identify them is RMATs. So it is basically a more advanced version of MATs. So for RMATs, it's able to analyze and replicate RNA-seq data. So here, because there's so many different types of alternative splicing, I'm just going to use one skipped exome as an example and to show what actually you can get from this analysis. Basically, here there are two different parts of the information I'm going to receive. So the first part is basically identify the location and the events where that actually happened. Okay, so basically it's going to tell where that happened, what is the upstream situation, what is the downstream context, where that skip happened. Okay, so the next part is more static analysis on the situation. So basically, for most cases, you're going to have two situations, two different types of isoforms. So one of the type is going to have this exome included, so it's exome inclusion isoform. And then you also potentially have another one as excluded or skipped isoform. Okay, and this basically is calculated 2 different situations and give you statistics on the situation about the events. So this is what you're going to get from the analysis. And then, so this is very difficult to see, it's a lot of numbers, but there's another way to actually visualize the results is through a sashimi plot. Basically, it's visualize the spliced junctions from aligned RNA-seq data and generate a gene annotation track. Okay, so this is compatible with IGV as well, so can visualize this using the platform I mentioned earlier on. So what is actually can see from this, so here it contains an event's name. It's going to assign a name, and then basically you have your mapped results. Okay, so and then also have your sample ID. So you can see those 2 are from the same situation, same group. Okay, probably more replicates, and then there's another replicates, and then they are different test conditions. Okay, and then it's going to have the junction reads number visualized for you as well. And then also there's a GFF file give you alternative isoforms. Okay, so this is the one basically has the exome included, and this one has the exome excluded, basically skipped. Okay, so this is a much easier way to see the splicing results. So besides that, we can also use RNA-seq data to identify fusion transcripts. So what is fusion transcripts? It is basically a merging of different fragment transcripts. So when this happens, usually it can be translated into a chimeric protein that could processes either a new or adapted function. So as you can imagine, because the function of the protein could change, that's why many fusion transcripts are actually classified as oncogenes that potentially have the potential to cause cancer, but also potentially contribute to tumor genesis as driver mutations. So it depends on how this is happened, the fusion transcripts where that actually happened. There's 2 different types. So if it's actually happened at a genetic level, basically the gene is fusioned itself, then this is a genetic fusion, genetic chimera. If actually the gene is not fusioned, it's only fused at the transcript level, then this is actually transcription-induced chimera. Okay, depends on how this is originated. Okay, and then the software we use to identify those fusion transcripts is called a star fusion. Okay. So basically, star fusion will take Illumina RNA-seq data as input and then generates a list of candidates of the fusion events as the output. So what does this software do? So here's the example. So here, basically, you can see, and this is transcripts M and N that is fused together. Okay, and then during the read mapping, and they will have 2 situations. So there are some reads it's going to encompass the fusion junction. Okay, and then there's also reads that actually spanning the junction here, and so sits at the junction exactly. Basically, star fusion just counts these 2 different type of reads. You can see here, and then tells you where this splicing, not splicing, sorry, and transcription fusion actually happens. Basically, the location of this fusion happened. What is where it's it has break at the left arm or under the right arm. So basically, the location of the events is described here. Okay. And then, so basically, so far we have used the eukaryotic mRNA sequencing results to went through the sequencing analysis workflow from quality control, raw data, starting with clean data. And then you have the second step, mapping to a reference genome, and then move on to expression analysis, identify differentially expressed genes, and then move on to functional analysis to assign biological function to those genes. And then on the other side, we have a characterization of the transcripts, including variant identification, alternative splicing analysis, and fusion transcripts analysis as well. Okay. However, in the next part, I'm going to talk about, there's other BI analysis for RNA-seq we can offer as well. So for the last part, we talked about a reference-based analysis for RNA-seq data, but actually we can also do the downward transcriptal assembly as well. Okay, basically, for this case, then there was no reference genome available, or this is incomplete. So then the RNA-seq data will be used to assemble and de novo into transcriptome. So one thing to pay attention to here is that if you wanted to go down this approach, the data required is usually much greater than a reference-based assembly. Okay, and so another BI analysis we can offer for RNA-seq is actually for PacBio iso-seq. So basically, this is a long-read-based sequencing approach. So I won't go into details about the difference between the two. Just wanted to highlight that for long-read cDNA sequencing or iso-seq, the advantage is able to capture many full-length transcripts, and then this would make the de novo transcript a lot easier, and also it's really good for identifying isoforms and fusion transcripts or other more complex transcript analysis. However, this is not suitable for expression analysis. So if you want an expression analysis, you still require the shorter read-based sequencing. This is really for de novo and transcripts and characterization. Okay. If you want to know more information, please again watch our previous webinar on this topic. Okay. So another analysis we can offer is actually for 10x based single cell RNA-seq and data. So for here, we'll be able to identify cell subpopulation. You can see here, and also we can analyze the marker genes detected, at different cell subpopulation and how they actually expressed at different cell subtypes. And also for 5' based experiments, basically VDJ aging analysis. So we can also offer the analysis for those as well. You can see here, this is just an example to review the frequency of VJ gene pairing across different samples. Okay. So another analysis we could offer for RNA-seq based experiment is metatranscriptome profiling. And so metatranscriptome profiling has become a very popular for study microbial community gene function and regulation at scale. So basically, we allowed researchers to understand the active function profile of the entire microbial community. So this is just a quick snapshot of the analysis we can offer. Basically, the first step, we're going to identify the transcripts, identify those unit genes, and use this information to reconstruct the metabolic pathways, basically. And then we'll be able to identify differentially metabolic genes expression in different community, basically. Okay. So in the end, I just wanted to summarize what we have talked through. So the major part is that we use mRNA sequencing example to go through our analysis workflow from quality control, mapping, and to quality expression, function analysis, and quantitative analysis. Okay, so this is basically shows you, if you have a bioinformatics analysis with us, what you're going to receive as a result. So you're going to have a report compressed file, .gc, so contains all the information. And once you unzip it, you're going to have a big package called results. So this basically contains all the analyze we just talked through. Okay, this is all included in the results package, and then all the images and the figures, and they're generated through them, through those analyses will be also included in them, and they are publication ready figures. Besides those, we also have a size check files and also a BAM file that can be used for inspect the mapping results. And also there are other files here that as reference genome and annotation, so they can be combined together to visualize together of the mapping results. And then we also, of course, will send you back the raw data. Okay, so as I mentioned, if it's a pair and a file, pair and a project, you're going to have 2 files for this. Okay, and then for the raw data, you can feel free to play around yourself, and then so this will be also delivered to you. And also, of course, there will be instruction to tell you, what is included, what is each file for. Okay, so that's basically bring to the end of the webinar itself. And I'm going to hand it over to our host, Rana.
Thank you, Shiki, for this wonderful presentation. We will now move forward to the question and answer session. I would like to remind all of you to put your questions in the icon that you can see below, which is marked Q&A, so we can answer all of your questions. So I will start with the first one from Irina, who is asking, how many samples are considered sufficient for differential gene expression analysis in the treatment trial with certain chemicals? She means by that treated and control groups.
So usually we would recommend 3 to start. So if you are aware, your sample, the variability of your sample is huge. Okay, then we recommend to increase it to 5. Okay, but usually a 3 is a good start point. Okay.
So 3 per condition.
Yes, yeah, 3 replicates for condition. Yeah, so that would be good. But you could sometimes also consider to pull technical replicates together, okay, and then just separate your biological replicates. So that way, sometimes can help as well. So, but 3 is the one we recommend as a minimal requirement. Yeah, but if the condition, for example, is a patient sample, or it's very difficult to control the background, they're going to be very variable, then it probably worth to increase the replicates number. Okay,
Thank you
that's
for
good.
your answer. Yeah, we have Konstantinos, who is asking, do you offer services for zebrafish RNA?
Oh, yes, zebrafish. Yes, we do offer zebrafish and RNA, but that's a very general question. There will be, as I mentioned here at the very beginning, you're looking at mRNA, or you want to know more about the or particular one. Are you interested in non-coding or a circular? So there's different solutions for that. But generally speaking, yes, for zebrafish RNA-seq, we do offer services for. Yeah. Thank you.
Thank you for your answer. We have Anna, who is asking if you know what program do we use for normalization?
So here is actually built in within DE-seq 2 already. Okay, so by default, okay, you will have you will have the FPKM. So that's the default. Okay, so you will have this data information for you. Okay, and also DE-seq has its own normalization built in as well. But by default, the normalization of reads, you're going to have this. Okay, but if you require other ones, we do offer TPM as well. So okay, that depends on your request and the project. If it's a single end read, it has to be RPKM, obviously. So yeah.
Thank you. We have Kristen, who is asking, she was actually wondering, how can RNA-seq compare to microarray outcomes, because the methods are different, but she is wondering if there are any studies comparing the differential gene expressions outcomes directly between.
I'm sure there are, because before the technology was developed for RNA-seq, previously it was all array-based. This RNA-seq is, of course, more dynamic compared to the range. It's going to be more dynamic because for the array, it's basically is color. If it reaches the saturation, then it can go even can be limited. And also array is predetermined. The probes, everything is designed. There's no chance for you to identify new novel transcripts. Okay, so but for RNA-seq, then you have the opportunity and to identify novel events and transcripts as well. And also, other analysis to characterize the transcripts, and so array won't have that function. But for expression-wise, yeah, so both method is fine. And then the major advantage is that it's not pre-designed. You don't need to know a lot, to design the panel, because the array is only available, has been designed for certain species, not everything. But mRNA, you don't really require that. You just need a high quality of total RNA, then you can sequence it. And then the range of the dynamic it offers is a lot better, it's more accurate. Okay, than the array. That's why the trend is moving, towards to using an RNA-seq approach. I hope that helps.
Thank you for your response. We have me who is asking, which sequencing depth do you recommend for differential gene expression for metatranscriptome analysis? This is the first part of the question.
Okay, so that's a really good question. So we have some information on the previous webinar, so please feel free, because this is really depends on the situation, how complex the community is. And do you think you're going to include your host information in there as well? Because that's going to require a lot more data to be analyzed. And then are you interested in something really rare presented in the community? So that means you need to sequence more depth as well. So I think the best thing to look at is to look for publications if there's any cases as there already. So then that would be the best guideline for you to start. And don't forget, you can always to add more sequencing afterwards. Okay, if you're worried or not sure, you can start with a more conserved, a little bit conserved data amount. Then you can add more sequencing after that. It is possible. Okay, so yeah, so best thing to do is think about your experiment design, how complex the situation is, and then look for related publications. And usually that's really a good start point for a project.
Yeah, so I guess you answered the second part of the question, which was, what is the depth you could recommend for a more detailed analysis or in-depth analysis? But I guess that we have to know
so that's really depends. You need to think about the complexity of the genome, and also what you want, what's the information you want to get. Do you want to just surfacely looking at expression level? So that can be slightly shallow. If you really wanted to identify some splicing ones, and then that's need to require more. And also if you target on different part of, if you are going to do more enrichment of the RNA you're looking at, or you're not going to do enrichment, you're just going to want to sequence everything, and the require on the depth to identify those rare transcripts, then that means the sequence needs to change depth as well. So there's quite a lot of things. So I think I would strongly recommend you to listen into our previous training. So that basically talks through the whole design of experiment and the things you need to consider at each step. Okay, so from your sample prep, to which type of library would be suit you, there's options, how do you choose different, services that suit you the purpose the best, also different platforms that you could use for your project, and some recommendations for the depth, very general as well. So I hope that helps. Yeah.
Thank you. We have Anna who is asking if it is possible to carry out total RNA sequencing from FFPE samples.
Yes, it yes, actually it is possible, but you have to possible wise it is possible. However, FFPE samples usually is highly degraded. Okay, and then the requirement for those library about those RNA, the quality usually is quite high. The amount you need to start with is quite high. So FFPE samples, some cases, okay, may not reach those criteria at the starting point. Okay, but if you have really nicely preserved or or samples and can generate a really high quality of total RNA to be used for this, and then it can be, as a starting point. So this is really depends on, the total RNA extracted, the quality and the amount. So not just the concentration, the amount, but also, the integrity of the sample. So you need to take that into consideration. So if you can reach that criteria, the answer is no. But from experience and most of the cases, so FFP samples has, a lot of, inherited problem with them, associated with them already. So there's a potential risk there.
Thank you for your answer. We have an additional question from Jade, who is asking if we can do metatranscriptome profiling on RNA-seq reads that have been taken from microbial culture. Just to give you a little bit more detail, he has I already have RNA-seq data from bacterial monocultures and co-cultures to look at the microbe interaction.
Yes. Okay. So that's actually something we can do. So we can deal with this, treat this as a bioinformatic analysis only project, and then the quick answer is yes. But we also will need to know, so what's the platform you sequenced, and then the data amount you have, and then do we, is there reference genome available, or actually we need to do de novo assemble? So it depends on the situation. So the quick answer is yes, we probably can do this, but we will need to have more information to really evaluate the project and then decide which would be the best approach for the project. I hope that address the question.
Yeah, thank you for your answer. We have come to the end of our question and answering session because unfortunately we have run out of time. So we hope that you really enjoyed our webinar and found it useful. I understand that we were not able to answer all of your questions, but don't worry, we have them all and we will come back to you very shortly with the response via email. And we strongly encourage you to contact your sales representative if you already know them. Here is a picture that shows you the sales representatives, or you can send us an email to info@novogene/europe.com, or send us an inquiry on our website and we will be delighted to reply to you. You will also receive an email link from us to the webinar recording and the slide deck. So this will be sent to you by Friday at the latest. I would like to mention to all of you that we are now offering the bioinformatic analysis for RNA sequencing, as you can see here. So what you can do is actually go on our website, www.novogene.com, and fill the inquiry. I would also encourage you to assist to our next webinar entitled DNA Sequencing and Its Applications. To our student attendees, I know that most of you require a certificate of attendance. So we are happy to announce that we have created a printable version of these certificates that you can access via the follow-up email. When you leave the survey, when you leave the webinar, a short survey will pop up on your browser. So we'd really appreciate it if you can take like 5 minutes to fill it, because this will really, your feedback really helps us to plan our next webinar topics. In the meantime, please stay connected with us. You can follow us on Twitter, on LinkedIn, or you can get in touch with us on our website, www.novogene.com, or email us at info@novogene-europe.com. We look forward to seeing you on the next webinar, and have a great day, and thank you.